Hello students, this is Dr. Amarpreet Kour, a lecturer from Jammu and Kashmir Education Department. Today, I will teach you about the development of embryo in monocots and dicots. Okay, the process is different in both of them. Before going ahead, I will suggest you to consult figure 2.13. There are two figures, figure A and figure B. Figure A, it shows a mature embryo sac, and figure B, it shows different stages of development of embryo. Okay, so consult first. The development of embryo from zygote is known as embryogeny. I will take embryogeny in dicot plants first. Firstly, we will start with a zygote, or you can say a fertilized egg. It divides into two unequal cells. One is a larger one, it is known as suspensor cell, and the smaller one, embryonal cell. Suspensor cell, it provides nutrition, support, and it also regulates the growth of embryo. Okay, the position of suspensor cell is towards micropyle, while as embryonal cell it is towards antipodal region. Okay, the suspensor cell, it undergoes transverse division and it forms 6 to 10 cell long suspensor. If you have any doubt in the transverse division, sometimes it causes a confusion between the transverse and longitudinal division. So for that, I will request you to consult the figure. Okay, so suspensor cell divides into and it forms a suspensor. The first cell of suspensor, which is towards micropyle, it is larger and it is called as historium. The function of historium in angiosperms or in other plants, maybe if it is present, is parasitic. It usually absorbs nutrition from other organisms. The last cell of suspensor, that is towards embryo cell, is known as hypophysis. It forms radical tip. Radical means a small root, embryonic root. Now embryonal cell, it divides twice vertically, that means perpendicular, and once transversely, and it produces two-tiered 8-celled embryo. That means now we have 8-celled embryo, 4 cells in one tier and 4 cells in another tier. The upper layer or upper tier is known as epibasal and the lower tier is known as hypobasal. Okay, epibasal tier, it forms two cotyledons and a plumule, while hypobasal tier, it forms hypocotyle and radical. Don't get confused. The last cell of suspensor cell, it only forms the radical tip, while as the epibasal tier of cells, it forms the real radical. Okay, now this octant embryo, that means 8-celled embryo, it undergoes periclinal cell division. What does that mean? It goes under cell division which is parallel to tissues. Okay, there are two types of cell division. One is periclinal and other one is anticlinal. Anticlinal means perpendicular and periclinal, it means parallel to the tissues. And finally it forms protoderm, procambium and ground tissue or ground meristem, and these tissues are responsible for formation of a well developed plant. Okay, these are actually the types, you can say, of the tissues. Okay, initially this embryo, it is globular, means like globe, But with the growth of cotyledons, it becomes a heart-shaped. You can see in the picture, it is very clear over there. Okay, in the final stage you will see there is a suspensor and a heart-shaped embryo. A dicot embryo, it contains two cotyledons, that means two leaves, and embryonal axis. Okay, embryonal axis, it is made up of two parts. One part it is known as epicotyle, and other part, or you can say the lower portion, it is known as hypocotyle. The epicotyle, it gives rise to stem tip, or the very small stem, that is known as plumule. Okay, epicotyle, it gives rise to plumule. And now the hypocotyle, it gives rise to root tip, or the embryonic root tip, that is known as radicule. Clear? Epicotyle gives rise to plumule and hypocotyle to radicule. Okay. Now, embryogeny in monocotyledons or in monocots. Here also the zygote, or you can say fertilized egg, it divides transversely and produces a suspensor and embryo cell. Okay, suspensor cell, it is having the same position as it had in dicots, means it is towards micropyle, while as the embryo cell, it is towards clausa. The embryo cell, it again undergoes transverse division and it forms top and middle cell. The top cell, it undergoes first vertical division and then transverse division and it forms a globular embryo. Okay, it is very easy in dicots. Sorry, in monocots. So first two divisions, they are transverse. The third division, it is vertical, and in final it is again transverse, and ultimately it forms a globular embryo. Clear? As the name suggests that in monocot embryo there is single cotyledon. That is why they are known as monocot. And this cotyledon, it is known as scutellum. At the lower end of embryonal axis, there is radicle and root cap. And this radicle and root cap, they are enclosed by a sheet that is made up of undifferentiated tissues. Differentiation means like formation of mature tissue, but undifferentiated means it is like meristem. Okay, and this sheet is known as coleorhiza. Clear? The shoot apex and leaf primordia, they are enclosed by a hollow kind of structure, and this structure is known as coleoptile. Okay, so the radicle, it is enclosed by coleorhiza, shoot, it is enclosed by coleoptile, and the cotyledon, it is known as scutellum. In some grasses, okay, there is remains of, you can say, second cotyledon. It is very small and it is known as epiblast. Clear? With this topic, I have completed the embryogeny in both dicots and monocots. So, thank you for listening to my lecture. In case you have any question or any comment, you can visit my website, that is www.amarpreetkour.com, and there you can post your queries and also you can listen to previous lectures.